In 2017, an article on CRISPR and the new field it entered in the year was published on ScienceNews.org, written by Tina Hesman-Say. The article highlighted the growing realization by the wider scientific and international community of the reality of CRISPR, a genome editing technology used to edit the DNA of organisms. While for the last 6 odd years there's only been acknowledged CRISPR editing done on animals, most authorities are fairly sure of activity in China with trials on human embryos.
Quoting ScienceNews.org, CRISPR is an editor consisting of a DNA cutting enzyme called Cas9 and a short piece of RNA that guides the enzyme. An enzyme is a substance provided by a living organism which acts as a catalyst to bring about a specific chemical reaction to a specific spot that scientists want to edit. Once the editing machinery reaches its destination, Cas9 cleaves the DNA. Now, until March of 2017, the editing of human embryos with CRISPR had only been a heated debate, but Liqian Tang of China's Beijing Proteome Research Center reported using the gene editing tool on a. Quoting ScienceNews.org, CRISPR is an editor consisting of a DNA cutting enzyme called Cas9 and a short piece of RNA that guides the enzyme, a substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction, to a specific spot that the scientists want to edit. Once the editing machinery reaches its destination, Cas9 cleaves the DNA. Until March of 2017, the editing of human embryos with CRISPR had only been a heated debate, but Liqian Tang of China's Beijing Proteome Research Center reported using the gene editing tool on a few viable human embryos, attempting to prevent disease-causing mutations.
Now a short history behind it. Francisco Manchica was the first researcher to name what is now called a CRISPR locus, reporting it in 1993. He did work on them during the 1990s and in 2000 recognized, as had been reported, that the disparate repeat sequences actually shared a common set of features, now known to be the hallmarks of CRISPR sequences. The very term CRISPR wasn't coined until 2002. In 2005, he reported that these sequences matched snippets from the genomes being the haploid set of chromosomes in a gamete or microorganism or in each cell of a multicellular organism, of a bacteriophage, leading him to the hypothesis that CRISPR is an adaptive immune system. The discovery and steps towards what CRISPR is today didn't stop with just the discovery of its existence, with significant moments in its timeline being in 2008 when Luciano Marafini and Eric Sotherman from Northwestern University in Illinois, America, discovered that CRISPR worked on DNA rather than on RNA, which was what was assumed. In December of 2010, Sylvain Monod from the University of Laval, Quebec City, Canada, used Cas9 to cleave targeted DNA, a major step in CRISPR's development, and in 2013 it was first used for genome editing, with Feng Zhang from the Broad Institute of MIT and Harvard McGovern Institute for Brain Research at MIT, Massachusetts, adapting CRISPR-Cas9 to be able to genome edit in eukaryotic cells, and demonstrated targeted genome cleavage in human and mouse cells.
Why is it such a monumental discovery? The discovery, further research, and our reality of the use of CRISPR/Cas9 is one of the greatest discoveries in science, but could have possibly the highest implications on humanity. Studies by biologist Kathy Niken of the Francis Crick Institute in London snipped DNA using CRISPR, intentionally creating a mutation in the human embryo to study the effects. The conclusions from the study and many others is that the gene editing tool is capable of making a variety of changes to the DNA, effects which stay with the individual for life and last for generations. Aside from the gene editing tool's simplicity, which is because CRISPR is able to be done when done onto an embryo, reaching and changing the desired genes much simpler and faster than other technologies. Jennifer Doudna, an American biochemist, in 2017 sat down with host Walter Isaacson at the Aspen Ideas Festival and talked about a couple of possibilities of using CRISPR.
start me with the animals. Give me a couple of examples like baby pigs or whatever that that science has already been able to use this to do.
Well, there's a lot of examples. So mice, that mice are used very commonly as models of human disease. So it's been possible to make mice that have changes to their DNA that make them more human-like in certain ways, make it easier to study the effects of therapeutics, drugs, for example, on genes. Similarly in, well, you mentioned pigs, so pigs, one of the attractive things with pigs right now is the idea of engineering them so they're better donors, organ donors for humans. And this is already being actively worked on both in research labs, but also in a startup company.
The possibilities of wiping undesirable traits or curing disease completely throughout humanity are tremendous. The DNA which is inserted by CRISPR is also progeny, meaning that it stays with the organism for the entirety of its life and lasts for generations further, being passed down by the parents.
Genetic engineering would open up room for discrimination and segregation. Those that are born with more desirable traits as a result of genetic engineering will be heavily favored in aspects of everyday life such as work, whereas people that have not been genetically engineered would face great difficulties. Rather than categorizing people by the content of their character and achievements, people will be judged by something they are born with and have no control over, their genes. The three most prominent areas of discrimination are all based off things people are born with and have no control over: race, religion, and gender. Genetic engineering will undoubtedly lead to discrimination. Unexpected side effects can be direct consequences of genetic engineering. For example, the editing of genes to create stronger bones might make your flesh weaker or your organs age faster. Essentially, editing genes in order to achieve a certain trait will also have the possibility of giving you unknown and unwanted side effects. Genetic engineering will heavily reduce the variation in the population, as people will all select the most in-demand and desired genetic alterations. As a result, people will be very similar in certain ways. For example, they might all have water-resistant eyes or more effective lungs. If something does go wrong, and a virus, for example, hits people with a certain gene, which is associated with water-resistant eyes, then this would be very detrimental to the general population. In a nutshell, genetic engineering will result in the majority of people sharing very similar genes, and if something goes wrong for one person, the chances are everyone else will be affected too.
After learning more about the topic, we still have some questions. One of these is how countries and governments would facilitate any sort of editing to our genes, either to the embryos or as alive humans. As a gleaming problem, maybe one country has a lot of editing done to its citizens, while another does not, leaving it disproportionately unfair internationally. Another question is what the consequences would be for those who either by choice of their own or guardians do not do any sort of gene editing and are so left with either undesirable traits or possibility to diseases. Would this lead to companies only employing those with improved bodies and create a second-class system? Our final question we still have unanswered would be how it would be done, either prenatal or waiting for consent from the individual. These are all major problems that the world must come together to discuss, and the reasons why it has been talked about so openly in the wider community, with scientists comparing the discovery only to that of the atomic bomb.